So you might not know this, but we live in probably the most extraordinary technological time in human history. So this is Moore's Law, which we all think about and know, essentially, is that as the pace of semiconductors, that your computers, your smartphones, all that power our lives have moved every 18 months and doubling in their capacity. But what's interesting is in DNA sequencing, the ability just to take an organism, grind it all up, and get all the DNA out and actually sequence it, the cost to do that for a human genome has precipitously dropped over the last 10 years. It's actually moved 3 times faster than Moore's Law since 2006. And so some of this technological change has actually enabled what we can call personalized medicine, participatory medicine. It's also enabled people to get excited about talking to each other and sharing together. There's websites like Patients Like Me where people say, I, for example, have Crohn's disease, and I've had a weird bacterial infection in my gut, and I've had it for 20 years, and I've had diarrhea for 20 years, and I'm trying all these various medications, but I have no idea if any of them actually work. But you can actually band together with other people and say, well, what have you been trying? What are the side effects? How many people evaluated it? You can have molecular data start to inform your own view of your own microbes and your own biology. And even at Cornell, when you're a patient at Cornell, you get blood work done, you can actually see what happens to all your microbial levels, different blood work done. You see this in real time when you log into your profile. And so my favorite thing about personalized medicine and precision medicine and thinking about all the genomes in us is that patients themselves actually enjoy it. So if you get quotes from emails from patients, they feel like they have access to their data just like they should. They actually get to think about their body in ways they've never imagined and monitor it for the rest of their life. Of course, it's not always clear, what it means all the time. But in some cases, this patient, when he had a high percentage of eosinophils, he had very itchy skin. So in this case, the brain is understanding different types of cells in its body and reinterpreting its own system, its own symptoms, and realizing, oh, this is actually okay because I was expecting those to be there. And in this case, an itchiness is somehow soothed by the knowledge of what caused it. And so this future that we kind of live in today is one of public and personalized medicine. So in this case, you could say that sharing really here is caring. People, myself included, have started to share all of their genomic data because essentially more data brings you more power. And so this applies to also other areas. And the second part is to think about citizen science. A lot of you are here because you'd be great citizen scientists, I think. So if you've never done molecular biology experiments, I encourage you to look at a lab in Brooklyn. It's called GenSpace. So it's a community laboratory. Say that you want to effectively join a gym membership. You can pay $50 a month, and here you can pay to join a lab, effectively. So actually, for some friends of mine, an economist, a sociologist, and a lawyer, I got them actually gift certificates to a one-day personal genotyping workshop. So you can go in, swab your cheek, and then test your own DNA. And yes, I did that because I'm a giant nerd. That's absolutely true. And I said, oh, it's your birthday. Well, we'll just give you this because I think I'd like to see you pipette. So most of their experiments failed. They did not learn to pipette correctly, so they didn't see any of their DNA. But that's okay. They actually learned a lot about how to extract DNA, to look at their DNA. And so when we were doing these experiments, I said to them, actually, if this was a different gene, you would not be able to look at this gene. So this is because a lot of genes up until recently were patented. So if you go back in the early 90s, certain genes started to become patented. So you couldn't look at this gene unless you owned that gene. And then over the years, in the late 90s, more and more genes got patented. And so we started to think about this. Actually, by the end of 2012, about 41% of all genes were patented, or even more, depending on how you did your threshold. So we challenged some of these patents as effectively a citizen scientist, as a scientist saying, wait a minute, I want to be able to grab my DNA from my body, anywhere in my body, and I want to be able to look at it, and you're telling me I can't look at it. That seemed a little weird to me as a geneticist. So actually I filed briefs and working with the ACLU to challenge some of the patents in 2009, 2010. What was patented was the gene and any copy of the gene, and even small fragments of the gene as small as 15 letters of ACGT. And if any of you have looked at sequences of DNA, that'd be like patenting the word because out of all of literature. And so it turns out every single gene in the human genome matches at least one other gene by 15 mers or even 20 or 25 nucleotide segments, or ACGT. So I'm happy to say that we challenged them. It went through the courts. There was questions about genomic liberty for all. It went all the way up to the Supreme Court. And actually, after oral arguments in 2013, I saw the director of the National Institute of Health high-five the chair of the Department of Genetics at Broad. We were very excited after the Supreme Court arguments. And indeed, actually, the claims were rejected. And just this week, some of the last litigation has fallen from Myriad Genetics. So I'm actually excited today to tell you that this idea of your free genome has started to happen. So I think what this gives you are certain... You have, and should always have, genomic rights. So this includes the right to look at your DNA, the right to have your doctor look at your DNA, the right to modify your DNA. And so this includes, so let's say, for example, you want to go to GenSpace and make GFP-positive cells. Like, say you want to make your own cheek cells glow green because you want to have a shower that glows green of your own cells. You know, I think that's your right. So you also should be able to PCR your genes, to look at them. That's just a way to amplify your genes. You should have that right. You should have the right to interpret your genome and all your omes, which is your transcriptome, your RNA, your DNA. And finally, I think if you want to, you can convert your genome to poetry. So I used a small Perl script to search for words in the English language that matched my BRCA gene, which infringed one of their patents, and just made it into this little haiku, which is very bad, but I'm also a bad poet sometimes in my life. So now you're like the Kool-Aid guy. Your genome is free, right? So this is fantastic. Oh, I'm excited to tell you today. 2 years ago, I'd be very worried, but today I'm very excited. So your genome is free. But that's the human genome where we spend a lot of time in my laboratory. But of course, there is more than one genome. There is something that all of you've probably thought about or heard about since you're here, is in this third section, the microbiome. So in your body, you are actually, if your body is a democracy, you are a minority party. If all the cells in your body were to have a vote as to where you should go walk, you would lose the election. They would win. In fact, it's actually a 10-to-1 ratio of bacterial to human cells in your body. So they outnumber you. And if you look at the number of active molecules of RNA, it's 150-to-1. They outnumber you. So they're everywhere. It's not so scary. But just to be fair, our cells are bigger, they're smaller, but they're still a 10-to-1 ratio. And so you start to think about this. If you look at human cells, they're big, but we're surrounded by bacteria. They're on our skin, they're in our GI tract, they're in our mouth, they're on all of our surfaces. There's many different kinds of them. But the big question is, what are they doing? Where do they come from? And so even in your own body, if you go to stand up on a scale, actually 3 to 5 pounds of your body is bacteria. Some of you more, some of you less. That's fine. You all look beautiful tonight. But a good chunk of your body is actually bacteria. And so there's a pioneering work in 2012 that looked across the diversity of the human microbiome, that different areas of the body are colonized by different bacteria and define to some degree health and disease. And so this includes all areas of the body that were studied tend to have their own specific niche effectively. And other interesting facts about the microbiome is if you look in your gut, actually 90% of your serotonin in your body and half of the dopamine is made actually in your gut and is sitting there. And so it makes you almost have 2 brains in your body. Some of you feel this way for other reasons, but there's maybe 3 brains in your body because a lot of it's in your gut. And so this is actually a critical component for mental health. So the Human Microbiome Project at the HMP and other various collaborations have actually shown extraordinary success in taking, say, microbiomes and enable or take away obesity from mice just by transferring the gut microbiome. You can alter the human gut microbiome in similar ways. It's been linked also to inflammatory bowel disease, and it's been linked to a large number of potential diseases, including cancer. So it's actually creating sometimes constant inflammation state. And this has led even organizations like Time to say, well, you are your bacteria. There's no more human cells. You are your bacteria. Why do they say this? Why is that true? It's because other factors of just the active molecules in your body, if you look in your bloodstream, about a third of them actually come from the microbiome. And this isn't just true while you're alive today, but it starts even before you're born. So mothers give us a lot, yes, besides love. Right before you're born, you have the placenta, which used to be thought as basically a sterile organ, but we now know it actually has a rich microbiome, and that the mother and fetus are constantly exchanging microbes and microbiota and microbiome during gestation as well as immediately after birth. Complication rates after vaginal birth are less than cesarean section birth, and breastfeeding obviously transfers antibodies, but as well as microbiome. And so in this case, this is another example where, sharing is caring, I'd say. So there's another case where sharing is caring, and this is, fecal microbiome transplants, which if you haven't heard of these, they are exactly what they sound like. So you take the fecal material from your gut and say, wait a minute, I have a C. diff infection. Again, I've had diarrhea for 20 years. I'll try anything, and you will. And well, since it worked in mice, I'll tell you what, let's just try it in people. And so the FDA began to say, wait a minute, patients are doing this themselves? Who's doing this? What's happening? And they said, you just go to thepowerofpoop.com And we'll help you, because here you can actually say, and by the way, 2013 was the year of the microbiome, the power of the poop. I mean, I couldn't make this up if I tried, but safe, accessible fecal microbiome transplant for all who need it. So the patients who are doing it themselves, participatory medicine, citizen science, all coming together, and people saying, well, this same thing, it's just sharing is caring. That's all it is. That's what's happening there. So this creates interesting policy questions as well. So is poop actually, is it a drug or is it a tissue? How do we regulate this from the FDA perspective? So Eric Alm's done great work in this space, and it's actually an ongoing question. I personally view mine as both, I would say. I think it's sometimes as a drug and sometimes as a tissue, but that's personal. This created sort of new policy questions. But the other factor that happens to most people is they start to think about poop, and they'll start to get scared, and then they'll go stay in their hotel room, and you'll see, oh, this is a clean remote because I'm at this Best Western, and this apparently is how they clean the remote because they're worried about what's left on the remote. I know some of you, maybe in this room, when you stay at hotels, I know some people bring sleeping bags with them because they don't want to touch anything in the hotel room because people are scared. And sometimes it's warranted, because if you go to the hotel pool, sometimes you'll see signs like this at the pool because you don't want fecal material in your mouth. I mean, people usually don't, except for the transplant people. In this case, sharing really is not caring, I'd say. So in this case, you don't want that. And if you go traveling in the airport, you'll see signs for avian flu, you'll see signs for Middle Eastern flu, different viruses that you should be scared of. And you almost feel like you need to leave the house in a BL4 suit because you're so terrified. So keeping this in knowledge and thinking about all the microbes around us and how they impact health and disease really brings it back to my daughter here, is thinking, what is she encountering every day when she wanders around the world? And most specifically, the first time I dropped her off at daycare, she's 6 months old, and she put her down on the floor, and she takes a toy and looks at it, puts it in her mouth, and is exploring the world with her mouth, as toddlers do. But then she put it down, and the next kid did the same thing, and put it in his mouth, and the next kid did the same thing. And so effectively, I realized they're all making out with each other through the toys, and I just thought, you know what I should do? Because the HMP papers had just come out. This is 2010, 2011, and I said, I should swab her mouth when I drop her off, and then come back at the end of the day, and I'll swab her mouth again, and see what's changed, but then I'd be that creepy dad swabbing all the mouths of the kids. So I thought, I can't do that, that won't work. But I kept thinking about other things, like, well, when she's with me in the subway, and she's still got her helmet on, she's grabbing things, what is there? What is in the subway, right? So this has led to a project that we launched in the lab where we just started looking around everywhere. So I have what you can call swab mania, and if you combine that with wanting to sequence everything, leads to a lot of interesting things. And I would actually add the list of rights that you have. A seventh one is that you have the right to sequence the DNA, I think, of any surface you touch. Because once you touch it, it can become a part of you. And this really creates, I'd say, surface-based rights. And so, which we engage with. So what is out there in the surface? That's why what a lot of you are wondering. So what we do know from other metagenomic studies, and metagenomic studies means you look at all of the genomes, bacterial, viral, mammalian, other ones, plants. There's been some work done from Craig Venter that looks at, say, indoor versus outdoor. The amazing things about some of the work that's known is the percentage of DNA. You take all the DNA and you sequence it from either inside or outside. It ranges anywhere from 25% to maybe even almost 70% of all the DNA comes from no known organism. We have no idea what it is. You look in the database of all known organisms, actually as Rob was alluding to, and we can't match it to anything. And they're right under our fingertips, and we have no idea what they are. And so, in this sense, it's been seen both indoors and outdoors, but even indoors, where you think we would be surrounded by bacteria that are normally with humans, a lot of them we don't know. And so, the hope was that actually a paper would be out, but it's still under embargo about some of the subway project. So I'm just going to mention a little bit the fact that we're launching all the data hopefully very soon. It's not all ready for release tonight because of we're waiting for the final steps of the paper. But I'll tell you that we're also going to try and engage more citizen scientists. So if you want to swab something, within hopefully about a week or so, we'll be able to actually enable you to take something, swab it, and join the festival of discovery effectively. So, and I'll tell you what else we're going to discover is other weird places. So the subway is weird, but it isn't as weird as this guy, who's actually collecting samples at the door to hell. This is from a crater in Turkmenistan. And here we're collecting samples from some of the hottest places and most acidic places to glaciers that are coming down from Greenland. We also have collected samples, some already from the deep sea brine pools at the bottom of the ocean. You have a lake at the bottom of the ocean called sea brine pools. And lastly, we're going to talk about is the space station. I'll talk about it at the end. We're going to actually be collecting samples from space, and we'll see how much of the DNA that's in New York City is actually alien DNA. So in the last two sections of this talk, I want to talk about the technology of what we're actually doing and how we're doing this and how more you can get involved. So most of the work we do today is with Illumina, where you actually look at surfaces and you can have billions of small fragments of DNA and you can sequence them all at the same time. This is what's called Illumina sequencing by synthesis. But what is on the cutting edge, what is sort of the bleeding edge today, something I'm telling you we're working on now, is what's called nanopore sequencing. So just about 6 months ago, we got into the laboratory a new way that we're thinking about sequencing in the subway in real time, not just some things that we collected a long time ago, but can we do it in real time? So the device is about this big. It can plug into your laptop, and it's a little tiny DNA sequencer. So what this sequencer is, it's built off nanopores. And so imagine if you have pores like you have on your cells, you attach them to a membrane which holds a circuit. And if you want to measure the current that's going through this integrated circuit, you watch as DNA starts to move through. And since you are measuring 5 bases at a time of that DNA, this creates 4 to the 5th states that you measure. And as DNA transits through the pore, you measure the current change. And what this lets you do is to measure the exact chemical presence of bases, modified or otherwise, as they move through the pore, much like if you were to slide through a big sock, essentially, and I could tell maybe that was you with your arm out or you with your head down if you're different bases. What it looks like on the output, and what we've seen from samples from the subway, is a little bit noisy right now. It may be beautiful, but it's a bit noisy, and here we call this squiggle space. So this represents the real-time transit of DNA through a single small pore. In our lab, we've even seen that you get many of them sometimes active. And what we can see now is at this point, even as long as a 71 kilobase, or 71,000 bases of a single strand of DNA that we've measured as it's moved through a pore. And so the reason I bring this up is because this is what I think will be in the future, and I'll talk about that a bit at the end, but that you could imagine doing this in real time, so that as molecules are moving through the air, for example, as people are worried about measles recently, or more historically even Ebola, there are ways to sequence DNA and RNA, because there's both kinds of viruses, directly as it comes through the air, and the future is almost here, I'd say. So I like sequencing, I like technology, but this creates a lot of big data. And so some people get very upset by the term big data. Some people feel like this. They get really angry because they say, I'm tired of hearing about big data. Everyone's talking about big data. Who cares about big data? But I'll make you care because my desk looks like this sometimes. And I thought this is just unsustainable. So instead of throwing them out the window, I said, we need a better way to organize this, but how bad can the problem really get? So if you want to sequence a human genome today, it's about 100 gigabytes. So if you have 1,000 of them, that's about 100 terabytes. That's not that bad, but what if all the 4.3 million new babies born next year had their genome sequenced? That's 430 petabytes. What if it was for everyone over 40? It could be 10 exabytes. And if you did everyone in China, that's 130 exabytes. And say you did every time you went to the doctor, you got your genome sequenced, and you did monitoring, and you did profiling. That could actually be 10 billion genomes with 10 time points. That's 10 zettabytes. And the interesting thing is that's just the human genome, which is here. That doesn't look at your DNA and how they interact with each other, your transcriptome, epitranscriptome, their modifications and changes, plus the epigenome and how DNA is wrapped around histones and their modifications, also nucleosome changes, gene regulation. Doesn't look at the proteome changes, the proteins that are moving through your body, or the metabolites, the small molecules moving through your body, and how they interact with each other, how our mRNAs degrade or transcription factor binding sites come in and bind things in your cells, or the database of all known annotations here, or your family medical history, or your bacteria and fungi, of course, and viruses in your body, each with their own genome, proteome, and transcriptome, and you compare this to drugs and vitamin levels and hormones in your body. And then if you compare that over time to the environment, before too long, you will get a yottabyte of data. That's a real word. It's 1 trillion terabytes. If you wanted to actually have that many data centers, you would have to move everyone out of Delaware and Rhode Island, maybe move them to Colorado. I don't know where we'd put them, but you wouldn't have enough space. Some people actually pronounce this yettabyte, but I prefer yottabyte, is what I prefer for the term. And so I'll remind you, your options, according to Yoda, there's really do or do not. There's no try, right? These are your options. So say you're like, I'm just going to do it. I'm going to just do it. I'm going to make a yottabyte of data. I'm going to make it. So all right, how much does that cost? I'm going to get my wallet out. You can get $100 for 1 terabyte. That's not so bad. All right, so what do I need? I need 1 trillion terabytes. It's a yottabyte. So I need $100 trillion. Well, how much is that? Here's the world's GDP, and here's the yottabyte cost. So you have to save for a while. That's not so bad. But the big question then becomes, all right, we can't sequence every genome at every time point and keep all of the data. It's just you can't do it. Okay, fine. So the big question then is, well, what to keep? And so I think the future really is again, I'd say in some ways it's starting to become here. And so it's very fitting that we're in this room because just about a year ago, we started a project on what's called the Twin Study from NASA to look at the genetic aspects of spaceflight. So I've been spending a lot of time more recently in Houston planning this mission, and where the plan is actually to send to the space station for a full year one astronaut who's an identical twin, Scott and Mark Kelly, and spend a year in space. We're going to be looking at the genetic information, how DNA gets mutated. We're going to be looking at structural variation and epigenetic information, how the DNA gets slightly modified at hydroxymethylation, methylation, and then DNA becomes RNA. See how RNA is expressed, how does it become active, and then methylated, what happens to the proteins, DNA, RNA, protein. Think about these protein changes. Think about how DNA is wrapped around different histones in the nucleus. Think about antibodies that are present after and before vaccination. Think about B cells, T cells, different cell counts. How long are your telomeres? We'll be assaying that. Look about cytokines. Think about targeted and global metabolomics. We'll look at the microbiome that's changing, of course, in the astronauts. Then looking at cognition, reaction time, and then vasculature. And do this effectively every 30 to 45 days for the next 2 years. And the collection just started about 3 months ago. So we actually will have, in this complete portrait of an entire human body, all of these changes. And the big question in my laboratory and in the field really is, which one of these is the most important? And the fundamental answer is, we don't know. No one actually has had this much data on one person over time, ever. And so it's exciting time because we'll start to look at this question of, can we actually send people to Mars in the year 2032, as what is the current NASA mission plan? Can they live in space for a year? And so we'll have some of the data to start to ask the question of, we hope so, or maybe, and try and mitigate some of the challenges that the body is not ready to deal with, from the microbiome down to the metagenome to other parts of the human genome. And the launch is actually for the astronauts on March 28th, and so I'm very excited the launch is coming. I finally want to end with thanking all the people in the lab who are fantastic and inspiring and work way harder than I do, and also teams that help for funding as well as for swabbing and collection in different aspects of all this work. And finally, thank you to the museum for hosting me, funding from the NIH, funding also from the museum and NASA, and a lot of great collaborators who are wonderful, and thank you for your time.